This is the first part of your photosynthesis notes where we introduce the players just like we did last time with cell respiration. So I'm going to go ahead and start. Alright, bottom left you have a plant cell and you got little green chloroplasts because they have chlorophyll in them. Up here in the top right you have kind of a schematic or a drawing of a chloroplast, and we'll talk all about the anatomy of that too. But big picture, we are going to make organic compounds, glucose, from light and air. We're going to use the energy in light, and we're going to use the carbon dioxide that's in there. And sound effects. So for an evolutionary historic picture, just a quick little look here. Units in the top are in millions of years ago, MYA. So if we have 4,000 million years ago, that's 4 billion years, right? The planet, if I drew another line right about here, or maybe a little bit further over, the planet's about 4.5 billion years old. So if we added that, that would go here. Our first evidence of life on this planet is actually about 3.8 billion years ago. So 3,800 million years ago, we have evidence of our first prokaryotic cells. Remember, these were doing glycolysis. This is an anaerobic process, but they did need to make energy. It wasn't a lot. It was only 2 to 4 ATP per glucose. So they were using simple compounds, and they were only doing glycolysis, right? This is 800 million years that passes. That's how long it took for the Earth to cool and for glycolysis, despite how complicated we think it is, to evolve. 800 million years. 800 million years. Another billion years pass before prokaryotes evolve the ability to do photosynthesis. Think about that. A billion years. You live 80, 85 years on average, humans do. A billion years pass. That's how long it takes for photosynthesis to evolve. When we have evidence that we finally have oxygen in Earth's atmosphere, because we don't have oxygen in Earth's early atmosphere here. There's no oxygen here in the atmosphere 3.8 billion years ago. Right, and we can figure this out. A lot of geologists actually can do this just by looking at rocks and metals in rocks, because you start to see red rock. You start to see the rusting of iron and metals in rock, and that's how we figured out how old Earth was when we started to see greater levels of oxygen. So these were this was occurring in prokaryotic cells called cyanobacteria. They were photosynthetic bacteria. And then here we go again. Another 500-ish million years pass before prokaryotes, I don't want to say figure out because they're not thinking, but I'll say evolve the ability to aerobically respire, to use the oxygen that their ancestors made a 500 million years earlier. They don't have mitochondria, but they do have a cell membrane, so all of the ATP synthase and a lot of those chemical reactions can still take place even though these are prokaryotic cells. About 500 million years after that, you have the engulfing of prokaryotic cells into eukaryotic cells, or sorry, larger cells, and this engulfing of an aerobic prokaryotic cell is going to provide compartmentalization, a specialization of structures that really kickstarts the evolution of eukaryotic cells. And the rest is history. And then here we are all the way down at the bottom. All right. Woo, sound effects. Let me turn those off. Turn them down. Big picture. Remember that your autotrophs, which include plants, algae, or protists, protista, and cyanobacteria, are going to make glucose. A lot of sound effects. Remember, and we're doing this, or they're doing this, by using the CO2 in the atmosphere. All of these molecules that are based on photosynthesis have CH and O. So everything else, all the nitrogens and phosphoruses, etc., that organisms have to consume or use to build proteins and phospholipids, etc., they have to come from the roots of producers, if you're a plant at least, right? These molecules, these organic molecules, are consumed and digested by heterotrophs that have to break them down, right? So consumers are kind of riding the backs a little bit of producers in the fact that they're consuming their organic molecules from the producers, but they are the same organic molecules that the producers made, right? So everything that you eat was made by a producer. And if you eat other consumers, then what they ate was made by a producer. Oh, there we go. There's the arrow. So remember back in cell respiration where we oxidized glucose. Right? We oxidized glucose. We removed the electrons because glucose is an excellent high energy molecule that can give away its electrons. It liberates ATP when you break these bonds. And we reduced that into a very low energy gas, carbon dioxide, right? Or sorry, we oxidized it. I keep saying reduced. We oxidized it. Oxygen gas became reduced, right? The electrons got added to oxygen, ultimately, because oxygen is very electronegative. It's an electronegative atom. It likes those electrons. And we reduced oxygen into water, right? The exact opposite process happens in photosynthesis. So here, we're not oxidizing glucose, right? We're not stripping it of its electrons. We're actually doing the complete inverse, and the formula is literally backwards here. We're taking CO2, a low energy molecule, and we are reducing it. We're going to pack it full of electrons to help build glucose. And we're oxidizing water. We reduce carbon dioxide and we oxidize water to get oxygen. Cell respiration released energy in the form of ATP. It was exergonic. Photosynthesis requires energy in the form of light. It's endergonic. Quick review from freshman bio, some structure. Remember, photosynthesis happens in the leaves. Right? Those are the solar panels of the plant. The leaves have little gates, these openings called stomata that open and close to let CO2 out, or CO2 in and oxygen out for gas exchange. Remember, glucose is just CHO. So any nitrogen, any phosphorus, any potassium, any other element besides carbon, hydrogen, oxygen, magnesium, we'll see that in a minute, all has to come up from the roots. So the roots are going to get you these other elements, and also the roots are going to uptake water. And that water, if you remember, moves up through structures called xylem. If you take a leaf and you zoom in, so here's our leaf here at the top, you zoom into it, you're seeing a cross section of the leaf, right? Photosynthesis happens in all of these cells. This is the spongy mesophyll, the palisade mesophyll. Here's your epidermal layers here. All of these cells that are green, they're green because they have chlorophyll, and that's where photosynthesis happens. Right? Notice that there's a lot of space in the spongy mesophyll so that you have diffusion of gas. And notice that you have stomata on the lower edge of the leaf to allow for gas exchange, and they open and close. Gas can come in and can go back out. One of the downsides, though, again, if plants had an existential threat, it would be desiccation, it would be dehydration, and that's because the higher water potential of the cells is going to want to lose water. Cells will lose water to the lower water potential air. Right? So cells and plants have a higher water potential than the surrounding air. So if they open their stomata too long, they risk losing too much water and drying out. So there's a cost benefit here. You want to keep them open to get oxygen out and CO2 in, but you don't want them open long enough that the plant dries out. This is why a lot of plants will close their stomata during the day and open them at night. And here's a a close-up magnification of stomata. You can see the two guard cells, and then you have the actual opening here in the middle. I'm going to skip. We're going to skip. All right, so let's look at the chloroplast in a little bit more detail. So many animations. I apologize. All right, so here we go. It is a double membrane structure. Remember, it kind of has its own cytoplasm called the stroma. A lot of chemical reactions will take place in there that we'll see in the next video. It doesn't have a folded inner membrane, but it does have, inside the inner membrane, it has these thylakoids, these disc-like membranes that are kind of stacked on top of each other. When they're all in a giant stack, it's called a granum. You don't really have to know that. We're not going to pay attention to that too much. We're going to focus more on the thylakoid membranes, because that's where all of these reactions are going to happen, and that's where photosynthesis takes place. Right. Remember that these thylakoid membranes, because they're doing photosynthesis, are going to contain chlorophyll and similar pigments. Pay attention to things that you've seen before. You will encounter the electron transport chain again. You will encounter ATP synthase again. You will encounter a proton gradient again. All of these things that you saw in cell respiration also show up in photosynthesis. If you look at the pigments really quick, so here we are in the chloroplast looking at just one thylakoid membrane. Again, the membrane it's a membrane, so it's going to be a phospholipid bilayer. But in that bilayer, you have embedded chlorophylls and other pigment molecules. And if you look at chlorophyll, you'll notice it's mostly carbons and hydrogens. There are some nitrogens in here. In the center, there's a magnesium, which actually makes this entire portion of the pigment hydrophilic. That's going to be facing the external, the outside of the cell membrane, or I'm sorry, of the thylakoid membrane. The inner portion, the portion that's going to be kind of in with all the fatty acids, is this, well, this kind of fatty acid looking tail. It has a lot of carbons and hydrogens, especially down in here. This is your kind of hydrophobic region. So this is anchored just like a protein would be anchored due to its phobicity or philicity. I don't know if I just made that up, but in the same way, this is anchored into the thylakoid membrane. There are many different chlorophylls. You have A, you have B, there are actually even chlorophylls C, D, and E. But really quick, one of the key differences is very, very simple groups that are attached to varying places. So for example, chlorophyll A has this methyl group here, right there. Chlorophyll B has a CHO instead. And that simple little difference changes the light that the pigment absorbs. And this is why plants have so many different pigments. Little teeny modifications like this provide a huge benefit. The more pigments, the more light you can absorb. It's kind of, if you break up the task of collecting light, you can collect more light. If you only had chlorophyll A, you wouldn't collect all the other wavelengths of light. So the more pigments, the better. But there is kind of an upper limit to the number of pigments because they are expensive to make. I'm going to let you look at this yourself. Just remember that we're dealing with the visible light spectrum here. Shorter wavelengths have higher energy. So down here in the violets and that would be ultraviolet, but your longer wavelengths, your reds, your oranges have lower energy. And the light that we see from plants, which is mostly going to be green, is actually because they reject that light. So let's look at a spectral graph really quick. Remember that if you are looking at a graph of absorption, which we are, we're looking at how much light the pigments keep. They take and they keep that, and they use it to split water and to form glucose. So here we're looking at two types of chlorophyll, A and B, And you can see that that one little chemical difference that I showed you before, right? The methyl group for chlorophyll A and the CHO group for chlorophyll B. That causes, even just a little teeny change like that, causes a pretty significant difference in how well these pigments absorb light, right? This is a pretty big shift for a little teeny difference like that in the kind of light absorbed. There's a little shift up here towards the higher wavelengths of light also, right? There are other pigments too. It's not just chlorophyll. You have carotenoids that absorb everything pretty much except your oranges and your yellows. So here's the carotenoid line, right? They do well with the violets and the blues, even some of the greens down in here, but then they don't reflect any of this, so that's why they kind of look orange. Xanthophylls will kind of look yellow. Anthocyanins will kind of look like a deep red or a purple color. So all of these different pigments exist. It's just that chlorophyll is the most common, and chlorophyll rejects green light the most. It absorbs it the least. So that's what you tend to see when you see plants. So that's why plants are green. All right, last slide here, and then we're going to finish. There are two main steps in photosynthesis, unlike the three in cell respiration. You have what are called light reactions, and you have the Calvin cycle, right? And look in the top right here. This figure is the same figure that's in your book. The light reactions are going to happen in the thylakoid membranes. The Calvin cycle is going to happen in the stroma, which is like the quote-unquote cytoplasm of the chloroplast. And just look at your basic reactants and products and where they are in this process, right? Light and water are going to be part of the light reactions. You're going to produce oxygen. CO2 comes in during the Calvin cycle, and we'll end up making sugar once that's done. Light reactions also produce NADPH and ATP. And when you recycle this, you recycle it as NADP+. So this is one of those things I want you to pay attention to. The electron carriers here are slightly different. They're not the same. In cell respiration, we had NAD and FAD as our electron carriers. In photosynthesis, we're going to have NADP as our electron carrier. The P, you can think of the P as standing for photosynthesis, even though it doesn't. It stands for phosphate. But if you see the P, it's kind of a cute little indicator that, oh, that's for photosynthesis, right? P for photosynthesis. So do pay attention to our different electron carriers. All right, we'll do that next time. This is the end of the first video. I hope that helped, and I will see you in video 2.